channel. So let us start with today's topic. Now till we are learning this lesson, what we will learn? We will learn what is how a sugar is tested, how starch can be tested, that in a food starch is present, how can we test that, how the presence of sugar can be tested, how the presence of fat can be tested, and how the presence of protein can be tested. So let us see with the first testing of sugar. Now here we will be doing this test with Benedict's solution. So this test is called Benedict's test. So what we are going to do? Now we can see that in a test tube some sample has been taken. So what is the sample? Now we are going to test about sugars. Now when in lab we are doing certain tests, if we are given some labeled test tubes like 1, 2, 3 and 4, if I am choosing the third number, we don't know whether it is a starch, a fat or a protein. So we need to perform each and every test to identify what is it. But now we are learning each and every steps of different tests separately. So we know that in this test tube we have already taken a starch or a simple sugar like glucose and fructose. Okay, so now when we are adding the Benedict solution in that, so we are taking, suppose we are taking a small amount of starch, any kind of starch we can take a crushed banana, we can take crushed rice. So we are taking and crushing it separately in a container, and we are adding this to the test tube. Then we are adding 2 to 5 drops of Benedict solution. So this is the Benedict solution that we are adding. So we are adding Benedict solution to it. Now we are heating it on a slow burner. So when it is being heated, now here we have taken that it is heated in a water bath. So if there is a burner over here and on top of that a water bath is kept, we are heating it, and what we are seeing that this red color precipitate is formed. So what is that red color precipitate? That is Cu2O or cuprous oxide. Now this shows the presence of glucose or sugar in this solution. Now however, if we are adding Benedict solution, in place of this, if we are taking tamarind, gourd or lemon, they do not show any red color solution after heating them in a slow flame or a slow burner, or if we are taking in a water bath. So strong heat is not recommended for this experiment. It should be heated in slow heat, and as we know that in lab we are generally using Bunsen burner and the heat is quite high, we can use a water bath also. So this shows that presence, as cupric oxide is the precipitate or the red colored material. So that we can see that if we are adding gourd, tamarind and lemon, this cuprous oxide is not coming as a precipitate. So this shows that they do not contain any sugar in them. So Benedict solution can only test the presence of simple sugars like glucose, and the compound sugars like sucrose cannot be tested by it. Okay, is it clear? So compound sugars like sucrose cannot be tested by it. We can only test simple sugars with the help of this Benedict solution. So this was a simple test for sugar. So is it clear to everyone? So the steps I have told, we are adding few drops of Benedict solution, heating it in a water bath or in a slow burner, then cuprous oxide is formed as a precipitate. But in place of this sugar solution, if we are taking any tamarind or lemon or anything else, this precipitate will not come and they doesn't have any glucose in it. Now the next test we will go for the starch. Now the presence of starch can be tested with the help of dilute iodine solution. Okay, so what we are taking? We are taking a sample method is, we will first take a sample in a test tube. We are taking a sample in a test tube. So we can test it, we are putting 3 to 5 drops of dilute iodine solution. Okay, this is iodine solution. Suppose this is iodine solution, we are placing it in the test tube, and we will notice that the color of the solution is turning to blue black after heating the solution. So this solution needs to be heated, and then we can see that a solution which is formed is turning to blue black, and this blue black colored substance, we see that this substance, as the result contains a blue black substance, we can say that the food sample contains starch. If on addition of iodine solution to a food there is no blue black color, we can say that no starch is present in the substance. So iodine is only responsible because it is only acting with the starch to make the blue black color. So combination of iodine and combination of starch gives you the blue black color. Thus any other food item will not give blue black color. So make it clear, very much clear, that when in any other solution, if we are adding this iodine solution and you want the color to come as blue black in lab, it will not happen. So when iodine drops are added to this food sample which was previously white in color, and if we are gradually slowly heating it and again cooling it down, then we can see that the sample is blue black in color. Okay, is it clear to all? Now the next one, we will be doing the experiment on fats. It is very easy example, and it is very easy one. So to test the presence of fat in different foods, what we can do now, it is the translucent spot test. Now we can see that we have taken two examples over here, same thing, only one in a paper and one in a cloth. So if we are taking some greasy substances, like if we are taking butter, ghee, mustard oil or anything, now if they fall on our clothes also, we have seen a greasy spot, or we can see some spot like substances appears in our cloth. So same thing we are seeing here, only thing is we are seeing it on some holding it on the top of a light. Okay, so if we are putting some oil in a drop of paper, or if we are crushing peanuts on a paper, or a what to say, in a cloth, white cloth, and we are placing that white cloth on a paper on a light, what we see, we can see a translucent greasy spot. So this translucent greasy spot which appears on the paper or on the cloth is due to the presence of oil, butter, ghee, mustard seeds. Also you can crush mustard oil, we get, right? So coconut oil, coconut also. So same thing here, we can see the testing of fat also. Sample of food is crushed here on the paper. Now we are rubbing the paper gradually, and we are holding it on the light to see the presence of the translucent spot. So this test is very easy. So the first test we did with Benedict solution, second test we did with iodine solution of for starch, and this one no solution is being added, just we can see it with translucent spot test. Now last we are going to do the test for protein. Let us see what we are doing there. Now here I have done the test of protein with the help of xanthropoietic test. So what we are doing here, let us discuss first. Now we are taking a small piece of food item to be tested. Anything can be taken, it can be hard boiled eggs or any protein substance. Now, and we are adding dilute nitric acid to cover the food item. Okay, so whatever we are taking, we are covering it with dilute nitric acid. We are heating the test tube very carefully as we are adding acid to it. So after a while we are noticing a yellow color in the test tube has come. Now gradually after the yellow color comes, we are rinsing off the nitric acid with water and adding ammonium hydroxide to it. And the color of the food item, that is the egg white, it changes from yellow to orange. Now this color changes from yellow to orange after we are rinsing it with water and adding ammonium hydroxide in place of nitric acid. Now this yellow to orange, it confirms the presence of protein in the food sample, and what you will see, you will also notice the same effect as we have done it. Suppose we have done it with the egg piece. Now we will see the same effect with peas and cheese because both contain protein. And this is because when the proteins are treated with dilute nitric acid, yellow color is produced, and if this solution is made basic, by adding ammonium hydroxide. So the acidic solution is made basic by ammonium hydroxide, and this color is obtained, and this test is called xanthroprotic test. And if we are repeating the process with a piece of apple instead of peas, cheese, or hard boiled egg, will you get this color? No, never, because apple piece is not a part of protein. You will find no yellow color on boiling the apple, and there is no question of making this solution again basic with adding ammonium hydroxide. So this is all for today. We have done 4 simple tests, one for starch, one for sugar, one for fat, and another one for protein. So I think this part is clear to all, and today by this we are ending this whole digestive system, or the digestive physiology ends here. Any questions, any queries, ask me, write me in the comment box so that I can clear it up in the next episode. Now we will come up with the next video, which will contain any other new interesting part which is related obviously to your syllabus. And the students, those who are not in ICSE, also you can subscribe to my channel, because these all are basic systems which are being taught in biology. So I have already posted many lessons in 10th standard. I am gradually posting the 9th standard lessons. Please do go through it, subscribe to my channel, and don't forget to share it with your friends. So more you subscribe to my channel, and you will get updates regarding whatever I am posting. And don't forget to share and write comments. So as of today, I am completing my episode. Thank you.